Hey, what's up and welcome back everybody to another video lecture of Royce Academy. In this lecture of today, I'm going to be covering on the topic of protein metabolism. So before going into the detailed explanation that how the protein metabolism operates in the human body or in the animal body, let's get into the detailed breakthrough of what the protein metabolism actually is and why is it so much necessary. So in case of protein metabolism, the first things I'm going to let you people know in the limelight that there are two basic pathways of the metabolism. These are the catabolism pathway and the synthesis pathway. In case of catabolism, the complex group of the proteins are being broken down into the simpler end products called the amino acids. And in case of the synthesis, the simpler groups of these amino acids are being turned over in order to form the complex group of the proteins. So why this catabolism and the synthesis functions in an allosteric mode of regulation or antagonistic or synergistic pathways. This is due to the fact that it helps in providing the energy, the bodily functions, the physiological regulations, certain other biosignaling aspects of the body, and many other biochemical and biosynthetic machinery that operates in the body of mammals and well as the human beings, specifically coming under the group of Animalia. Now, this protein diet, why is this protein necessary? In which cases, in which specific creatures, this protein metabolism is very much vital and necessary or is more prevalent? In case of the animals and the mammals, which are humans and the mammals, which are coming under the group of Animalia, we know that we consume protein-rich diets like that of the meat, fish, eggs, cheese, milk, curd, yogurt, all this like milk protein substitutes or plant protein substitutes, also animal protein substitutes, all these things that we consume on a daily diet basis, being omnivores and also the carnivores who consume only the meat or the meat proteins or the animal proteins, and the herbivores which consume the plant proteins. In bodies of these type of creatures, the protein metabolism is very much necessary in order to have the end product of the amino acids and also the protein synthesis from these amino acids in turn during the time of emergency in order to have the vital physiological and regulatory or signaling aspects that is very much necessary for the proper functioning, growth and development, energy buildup of the body. So in case of protein metabolism, we come across two specific pathways that how the catabolism and the metabolism synthesis happens. In case of these two pathways, both of these are directed through two types of synthesis. One is the de novo pathway and another one is the salvage pathway. Now I'm going to let you people know one thing here, that this de novo and the salvage pathway are basically the pathways which are necessary for the biosynthesis of the nucleotides. What are these nucleotides? The nucleotides are the components which are having the ribose 5-phosphate, or the sugar or the 5-carbon sugar, and also the phosphate group, and also the nitrogenous base. So in order to synthesize these nucleotides, that is the pentose sugar or the ribose sugar, that 5-carbon sugar, and the phosphate group, and along with the nitrogenous base, this biosynthesis of the nucleic acids through the de novo pathway is much important. There are two pathways, de novo and salvage pathway. Now what's this de novo pathway is basically? The de novo pathway deals with 4 different kinds of molecules which are very much necessary to direct these pathways. I'm going to cover those pathways, 4 different things and details of those lectures or topics in my lecture ongoing. But before that, I'm going to tell something about the salvage pathway. What happens in the salvage pathway is the salvage pathway is actually a directed pathway which happens to be happening with the intermediates of the amino acids that have been formed from the in byproducts or as a secondary products from the de novo pathways. So in the case of emergency or in time of emergency response where the protein is very much less in a body, the salvage pathway builds up when the de novo pathway is malfunctioning or not happening properly, or if it's happening properly, it is not providing the adequate amount of protein that the body needs. Right. So in case of the de novo pathway, let's have a look on those molecules. What are the important molecules that are present? In case of de novo pathway, we have amino acids, ribose, 5-phosphate, CO2, NH3. Okay, so you see these 4 molecules right here. So from where did this amino acid, carbon dioxide, ribose 5-phosphate comes? These amino acids are mainly coming from protein diet. protein diet that we consume on a daily basis, like I said, people. And this ribose 5-phosphate comes from the carbs diet, right? And this CO2 molecule comes from where? Cellular respiration, from where the C3 glycolysis and the Krebs cycle happens. And this ammonia group is being directly transcribed from the amino acids. Right. Now, the thing is, all these four molecules are very much necessary for operating the de novo pathway. In this lecture today, I'm going to be covering on the topic of purine pathway, basically, and how the purine de novo synthesis of nucleotides, purine nucleotides, the major two master nucleotides, adenine and the guanine, happens in the de novo pathway. Right? It's a huge pathway, basically. So, these amino acids, what are these amino acids? These amino acids are basically of two types: glutamine and glycine and aspartate. Now, this glutamine is very much necessary as it helps in incorporation of NH3 within the biomolecules of the cell. All these pathways are happening within what? Within the cytosol, endoplasmic reticulum, and to somewhat Golgi complex, Golgi bodies. Okay, so this is where all these pathways are happening, more or less. Now in this case, this glycine and aspartate also helps in the functioning of incorporation of the amino acids as an intermediate within the pathway of the de novo synthesis. Right. So in the purine nucleotide synthesis through the de novo pathway, or in the pyrimidine nucleotide synthesis through the de novo pathway, all these three amino acids are very much important or plays a crucial role in incorporating the ammonia group within the biomolecules, which thereby themselves cannot incorporate or thereby cannot associate the ammonia group. So this nucleotide synthesis is very much necessary because with the formation of these nucleotides, it helps in transcribing the structure of the entire DNAs or the RNAs of the nucleic acids, which is very much necessary for the cell maturization, proper cell formation, functional cell formation, and also helps in other cellular functions of the body that directs the physiological regulations, the tissue functioning of the body, the bioclinical aspects of the body, and other each and every cell signaling that helps in a proper functioning and regulatory aspect of the body. So it also helps in the replication and formation of newer cells, synthesis of proteins. So the synthesis of nucleotides is very much necessary by the de novo pathway. So all these topics are covered under the pure protein metabolism aspect. Now, you see this ribose 5-phosphate here? Where does this ribose 5-phosphate come? The ribose 5-phosphate cannot be directly taken up or incorporated. The ribose 5-phosphate cannot be directly incorporated or taken up by the molecules within the endoplasmic reticulum or cytosol in order to initiate the process of the de novo pathway. So the ribose 5-phosphate has to be converted into some other forms. The ribose 5-phosphate in presence of Mg2+, an enzyme, ribose phosphate pyrokinase, okay, helps in the formation of 5-phosphoribosyl, 5-phosphoribosyl 1-pyrophosphate, that is PRPP. Okay, so this 5-phosphoribosyl, you see this compound right here, 5-phosphoribosyl 1-pyrophosphate. This compound is what it initiates. This compound is what that initiates the process of de novo synthesis of the purine nucleotides. And this compound is being delivered or liberated as an intermediary product of several other protein metabolism pathways that happens within the hepatocytes of the liver cells or of the muscle cells in order to build up the actin and myosin proteins, which are basically needed because most of the proteins are needed in case of the muscles, and few proteins are needed for the formation of enzymes and the signaling molecules which are necessary for the physiological regulatory aspects of the body. Now, let's get into the fact that how the CO2 is coming. CO2 is coming from the glycolysis, Krebs cycle, and the C3. So CO2 is necessary for the carboxylation and decarboxylation for the formation of several other intermediary compounds, or the compounds which are crucial for the direction or direction of the pathways towards the final product or the final compound of the purine nucleotide. And this ammonia is being liberated in order to assist the formation of the nucleotide, complete nucleotide chain or complete nucleotide ring. So now let's get into the fact that how this pathway is operating. Now let us start with the molecule 5-phosphoriboside 1-pyrophosphate PRPP. In this case, what happens is this molecule initiates the process of the de novo synthesis, like I said. So this molecule in the presence of Mg2+ ion, also in the presence of glutamine, which gets converted to glutamate, right? In the presence of the activating enzyme called glutamine synthetase. Glutamine synthetase helps in the activation of inactive glutamine to the activated to glutamate, and the inactive glutamine synthetase gets activated to active glutamine synthetase in the presence of the energy that is being benefited by the conversion of ATP to ADP plus PI. So it's an active process, energy seeking process, where the inactive glutamine synthetase is getting converted to active glutamine synthetase, which is thereby converting the glutamine to glutamate and dropping off or warding off the amine group, which is being transcribed with this 5-phosphoriboside amine. What happens in this case is this reaction is catalyzed in the presence of glutamine PRPP amido transferase. Right. What happens in this case is you see this glutamine group, amine group right here. This amine group is being directly incorporated with the ribosyl, and this pyrophosphate group that is PPI drops off. So the phosphoribosyl gets incorporated with the amine group, so 5-phosphoribosyl amine it's forming, right? Now this 5-phosphoribosyl amine in the presence of glycine, which is another amino acid, and with the conversion of ATP to ADP plus PI. It's an energy seeking process in the presence of enzyme glycinamide ribonucleotide synthetase helps in the formation of the glycinamide ribonucleotide GAR. Now this GAR molecule in the presence of formyl group, okay, transformylase, this glycinamide ribonucleotide, this molecule in the presence of N10 formyl H4 folate gets converted to H4 folate, and this happens in the presence of the enzyme known as GAR transformylase. Helps in the formation of what compound? This formyl group. You see this formyl group right here? This formyl group. This formyl group is being attached with the GAR. This formyl groups get attached with the glycinamide ribonucleotide, which helps in the formation of N-formyl glycinamide ribonucleotide, which is also called FGAR. Formyl glycinamide ribonucleotide. Now this FGAR complex, in the presence of FGAR, again this compound, what happens in this cases, it takes help of another molecule of glutamine, which gets converted to glutamate, along with the conversion of ATP to ADP. ATP to ADP plus PI in the presence of enzyme FGAR amido transferase. Again, in this case, what happens is this amine group from the glutamine, this glutamine molecule, this amine group donates its amine group to the N-formyl glycinamide ribonucleotide, and glutamine itself gets converted to glutamine. This is also an energy-seeking process, so ADP gets converted to ADP plus PI. So this is an active process. So the end product of this catabolism is formyl glycine amidine ribonucleotide, which is also called FGAM. Okay, what happens is this glycinamide is already having an amine group with an amino acid that is glycine, okay, which was incorporated in the second step. Now with this glycinamide, in the amine position, again another amine group is being attached. So it is forming an aminidine group. This group, the aminidine, aminidine, the aminidine, because it is having two amine group attached with the same molecule at the same position of the ribonucleotide chain or the nucleotide chain. And now what happens is this FGAM, in presence of another enzyme called FGAM cyclase or AIR synthetase, okay, helps in the formation of 5-amino imidazole ribonucleotide. AIR. Right. So what happens in this case is this formyl amino ribonucleotide helps in the formation of the amino-imidazole ribonucleotide, where the formyl group is being removed within the presence of enzyme cyclase. Or, in the other hand, the AIR synthetase, that is amino ribonucleotide synthetase, helps in the formation of this 5-amino-imidazole ribonucleotide from the body. What happens in this case is this cyclase molecule or the synthetase molecule, amino-imidazole molecule, removes the formyl group. You see this formyl group right here? This formyl group and the glycine is being removed from the ribonucleotide chain, and helps in the formation of amino imidazole. It's imide group and amino group attached together, despite or irrespective of the formyl group, which is being warded off or kicked off from this compound and gets converted to 5-amino-imidazole ribonucleotide. So here, the amynidine is getting converted to amino-imidazole in the presence of the cyclase or amino-imidazole synthetase. So this glycine amynidine compound is getting converted to amino-imidazole compound, where the glycine molecule is being removed partly, and this amynidine is getting converted to imidazole group in the presence of FKM cyclase or AIR synthetase. Now this is also an energy seeking process where ATP gets converted to ADP plus PI. Right? ADP plus PI. Now this 5-amino-imidazole ribonucleotide AIR, this 5-amino-imidazole ribonucleotide AIR in the presence of again an ATP molecule which gets converted to ADP. This is an energy dependent process again. ADP plus PI. Most of these processes are the basic thing is, in most of these conversions of the reactions, most of these processes are all energy dependent, because without the energy dependent, for incorporation of a new molecule or the transport of a new molecule within the cell and incorporation with an existing biomolecule is not possible. So in this thing, you can have a question always in your back mind that why this ADP is converted to ADP plus PI always. This is the only reason that why it's getting converted, that ADP to ADP plus PI is getting converted deliberately all the time when a newer molecule is being formed. So this 5-amino-imidazole ribonucleotide, with the incorporation of the energy that is being liberated or that's been cut from the ATP to ADP plus PI conversion in the presence of an enzyme called CAIR aminotransferase, CAIR synthetase helps in the formation of N-carboxy amino imidazole ribonucleotide. CAIR. Okay, carboxyaminoimidazole ribonucleotide, the ADP to ADP plus PI, right? CAIR synthetase, the carboxyaminoimidazole ribonucleotide, CAIR. Now, this CAIR, the carboxyaminoimidazole ribonucleotide, is again getting converted to another compound, which is the same, almost like this, in the presence of enzyme CAIR mutase, which helps in formation of carboxy amino imidazole ribonucleotide. That is CAIR again. Now what happens in this case? Why am I writing these two compounds together, two compounds repeatedly, right? You see this N-carbamino-imidazole ribonucleotide CAIR. In this case, what happens in the chain of the ribonucleotide, in the particular position where that amino-imidazole group is being attached, that is, one amine group and the imide group is being attached together, in the same position the carboxy group is present. But in this case, what happens in the presence of mutase enzyme, the carboxy amino group is being transferred to another carbon of the ribonucleotide structure, ribonucleotide ring, whereas the imidazole or the imide group along with the ol group, imidazole group, is being attached to another position, another carbon of the ribonucleotide chain. So basically, this is nothing but the transfer and the rearrangement of the carboxy amine group from one place position to another within the ribonucleotide chain. Okay, now this conversion can be directly assisted from this 5-amino ribonucleotide. This conversion can be directly assisted to the formation of the carboxyimidazole in the presence of an enzyme called AIR carboxylase. Okay, now this is not an energy dependent process because only the rearrangement is happening, right? Only the rearrangement of the atoms are happening here. Now in case of carboxyimidazole ribonucleotide, this CAR compound in the presence of another molecule of amino acid, basically aspartate. Aspartate helps in the formation of the cycar. Cycar is N-succinyl comma 5-amino imidazole 4-carboxy ribonucleotide, which is called SAICAR, cycar. Now this cycar molecule, how does it happen? This cycar synthetase in the presence of the enzyme cycar synthetase, along with the help of the energy that is being acquired by the conversion of ATP to ADP plus PI, this cycar complex is being formed. Now the cycar complex, what it does is, this cycar complex with the liberation of fumarate and presence of enzyme, with the help of enzyme cycar lyase. Now what is the function of this lyase here? You see this succinyl group right here. This succinyl group in presence of the lyase gets converted to fumarate and wards off or kicks off from this compound. So only the 5-amino imidazole 4-carboxy ribonucleotide ICAR remains. So which helps in the liberation of the 5-amino imidazole 4-carboxy ribonucleotide. ICAR. Okay, now this ICAR molecule, again with the incorporation of a formyl group. Now why does so much of the cofactor, so much of activity is happening for the synthesis of a nucleotide? It is not that the nucleotide is going to be synthesized simply by combination of different amino acids one after another and through some basic formilations or acetylations or methylations, addition of the HCH group or CH3 group or CH2 group, whatever it is, or wall group, a nucleotide is going to be formed. But in this case, what happens instead is the nucleotides are being formed in a very complex and complicated manner with the sequential addition and signaling of the chain of the formation of the nucleotide, and all of these processes that one after another, when the formylation will happen, when the succinylation will happen, when the removal of the carboxyl group would happen, when the carboxylation prior to that will happen, is always cell signaling. So this amino acid, like I said, the proteins, the intermediary products that are being broken from the other protein metabolisms or the catabolism pathway, these amino acids which helps in the formation of other amino acids, which helps in formation of other proteins, are signaling continuously those molecules which are important for getting incorporated within the chain or within the ribonucleotide operation or the pathway process in order to form a mature ribonucleotide ring. that is very much necessary for transcribing the structure of the nucleic acids, that is DNAs and RNAs. Now this 5-aminoimidazole 4-carboxyribonucleotide ICAR in the presence of again the formyl group, where N10 formyl H4 folate, which gets converted to H4 folate, and in the presence of the ICAR transformylase, the enzyme here is called ICAR transformylase. Let me increase the arrow mark so that I can write it within the latent portion. ICAR transformylase. Again, here what happens is just the same step which happens here. The formyl group that has been present in the N10 formyl H4 folate is being transferred to the 5-aminoimidazole 4-carboxyribonucleotide ICAR, which helps in the formation of N-formyl 5-aminoimidazole 4-carboxyribonucleotide. Which is called FAICAR. FICAR. Okay, FICAR. This is FICAR. FAICAR. FICAR means formyl 5-aminoimidazole 4-carboxyribonucleotide. Now this FICAR molecule in the presence of inoxidase or inoxidase monophosphate synthetase, along with the dependence of the acquiring energy from this FICAR molecule in the presence of IMP synthetase, helps in the formation of inosine 5 ribose monophosphate. Okay, so this compound right here, inosine ribose 5 monophosphate, you see this compound right here. This compound is the first stable precursor of the nucleotide. It's not even the nucleotide right now, because the nucleotides are adenine and the guanine, as we all know in the Purine cases. This is actually the precursor of the adenine and the guanine nucleotides that are being formed from the formyl amino carboxyribonucleotide. Okay, formyl, that's the FICAR group. So from this is an energy seeking process again. Here also the ATP gets converted to ADP plus PI. Right. So let's have a recap up to here. This 5-phosphoribosyle, like I said, 5-phosphoribosyle is coming from the ribose 5-phosphate plus ATP in the presence of Mg2 plus and ribose phosphate pyrokinase. That's the enzyme which helps in the formation of 5-phosphoribosyle 1-pyrophosphate PRPP, which is an intermediary product of several other amino acid or protein metabolism steps like synthesis or catabolism that happens in the hepatocyte cells of the liver or the muscle cells. Okay. Now, in case of this 1-pyrophosphate PRPP, this gets converted in the presence of Mg2 plus with the incorporation of the glutamine, which helps in donation of the amine group or an H3 group, and gets converted to the glutamine, and that glutamine is getting converted to glutamate in the presence of activated glutamine synthetase. Now, this glutamine PRPP aminotransferase helps in transferring this amine group to the 5-phosphoribosyle part, so it forms 5-phosphoribosyle amine, which again in the presence of glycine, which is another amino acid, glycinamide ribonucleotide synthesis, with the incorporation of the energy from the ATP, helps in the formation of glycinamide ribonucleotide. Now this GAR, in the presence of GAR transformylase with the addition of the formyl group, the formyl group which is coming from the N10 formyl tetrahydrofolate is being incorporated as the N-formyl glycinamide ribonucleotide. Now, why does this formyl group gets attached in the formyl group? Why is this step necessary for the addition or incorporation of the formyl group? This is because the formyl group, which is getting formylated at the end position of the particular glycinamide, where the glycine and the amino acid is being attached with the ribonucleotide chain, it helps in signaling or helps in identifying the molecule. It helps as an identification mark of the ribonucleotide molecule, so that the further amine or the further amine groups or the further processes of all the sequences and the steps can easily happen. So all these other molecules or enzymes can identify the formyl group, and it's like something like happening in the capping and the adenylation, like in case of translation. In translation, we know the 5' end is undergoing capping and the 3' end or the tail is undergoing adenylation. The capping position is undergone because it helps in the spliceosome enzymes, the spliceosome complex 1, 2, and 3, to identify the heteronuclear mRNA and do the splicing reactions. Now, the polyadenylation helps in maintaining the stability of the mRNA while the splicing is going on. Right. So formyl group is here nothing but acts as an identification mark for the other subsequent enzymes that helps in converting this FGR molecule to the final nucleotide, that is inosine monophosphate. Now that's why the formylation step is much important and necessary for this aspect. Now the FGR aminotransferase, in the presence of glutamine and glutamate, and in the presence of enzyme, forms FGAM, the formyl glycinamide DIN ribonucleotide. Now the amine group already existing with the amine helps incorporation of another amine group, which helps in formation of aminidin group. Now this aminidin, with the help of the FGM cyclase and AIR synthetase, forms the 5-aminoimidazole ribonucleotide. Now the 5-aminoimidazole ribonucleotide, in the presence of CAIR synthetase, forms N-carboxyaminoimidazole ribonucleotide, which is again in the formation of the acquirement incorporation of the energy from ATP to ADP plus PI. Now this CAR, in the presence of CAR mutase, CAIR, it gets converted to carboxy aminoimidazole ribonucleotide. What happens in this case is the same carboxyamino group is being detached from this imidazole group at the same particular position of the amine group of the ribonucleotide chain, and gets transported to another, or repositioned to another position of the nitrogen that is present in the ribonucleotide chain. So basically this is not an energy dependent or passive process, because in this case what happens, rearrangement of the atoms are happening. So but this process can be directly transcribed with the presence of enzyme CAIR synthetase, in the presence of enzyme AIR carboxylase, where the carboxylic group is being rearranged directly from the 5-aminoimidazole ribonucleotide AIR to the carboxylase group carboxyaminoimidazole ribonucleotide. Now the CAIR, in the presence of another energy dependent process, in the presence of enzyme cycar synthetase, forms N-succinyl 5-aminoimidazole 4-carboxyribonucleotide cycar. Now the cycar leaves or kicks off the fumarate from the succinyl group in the presence of cycar ligase. What does ligase do? It breaks down the molecule. As we know that ligase joins the molecules, and ligase helps in splitting down complex molecules to the simpler ones. So the succinyl group is being removed in the form of the fumarate group, and the fumarate, this compound is again getting converted to 5-aminoimidazole 4-carboxyribonucleotide, that is ICAR. Now this ICAR molecule, in the presence of again the formylation is happening here, in presence of ICAR transformylase, helps in formation of N-formyl 5-aminoimidazole 4-carboxyribonucleotide, that is FICAR. FICAR, in presence of ADP, which donates energy in the process, and IMP synthetase in this final stage, which helps in identifying the formyl group of this entire 5-aminoimidazole 4-carboxyribonucleotide of this entire molecule, helps in identifying the formyl group and incorporates the inosine ribose 5-monophosphate. That is the nucleotide, the inosine 5-monophosphate, that is the first precursor or the stable nucleotide ring chain structure that is being attached with the 5-aminoimidazole 4-carboxyribonucleotide. Now this IMP molecule, that is inosinate ribose 5-monophosphate, is acting as, or actually is the checkpoint molecule of the entire de novo synthesis of the purine nucleotide pathways. What did I tell you about the purine nucleotides? There are two parent purine nucleotides majorly, the adenine and the guanine, in case of both the nucleic acids, that is DNA and the RNA, right? So from here, Adenosine 5 monophosphate directs two pathways basically. It splits up into two pathways. In one pathway, in the presence of enzyme adenylosuccinate synthetase, and in the presence of the incorporation of energy from GTP to GDP plus PI, it helps in the formation of adenylosuccinate, adenylo ribose 5 monophosphate. Now adenylosuccinate, this compound, this particular compound, again in the presence of adenylosuccinate lyase, helps in kicking out or removing fumarate and helps in formation of adenylate ribose 5 monophosphate or AMP. Now this compound here, you see this compound here, this particular compound is one of the two Purine nucleotides that helps in its formation, or that gets converted or formed in the de novo pathway from the IMP checkpoint. Okay, now why am I calling this IMP as a checkpoint? I'm going to explain you later. First, let me do the other nucleotide. Okay, now this IMP in the presence of IMP dehydrogenase and with the help of NAD plus, which is accepting the hydrogen in the form of electron and converting to in the form of hydrogen and converting to NADH plus H plus in the form of colon H plus E minus. Okay, in that way it is helping in the formation of the xanthylate ribose 5 monophosphate or XMP. Now this XMP compound in the presence of again another molecule of glutamine, which gets converted to glutamate in the presence of XMP glutamine amido transferase helps in the formation of guanylate ribose 5 monophosphate or GMP. And now this also takes place in the help of energy dependent process converting ATP to AMP plus PPI. Now this, you see this compound right here, GMP, this is the second most important compound of the Purine nucleotides that have been formed from this checkpoint of the IMP. Why does this happen anyway? Because IMP, inositol ribose 5 monophosphate, from here it acts as the allosteric feedback regulation. Right. In this case of allosteric feedback regulation, what happens is the IMP monophosphate acts as a checkpoint. The IMP liberates the formation of the guanylate monophosphate and the adenylate ribose 5 monophosphate, GMP and AMP. Now when in the cell, suppose in the cell cytosol or the endoplasmic reticulum or in the Golgi complex, the GMP and AMP concentrations are too much high. Those two molecules act synergistically or contrarily or conversely in order to direct the partial inhibition of the phosphoribosyl pyrophosphatase conversion up to the IMP. So the first pathway, that is the conversion of IMP from the PRPP, the larger pathway which I've just shown right now before this pathway, is being partially inhibited synergistically, that is together in an amplified action due to the presence of GMP and the AMP compound, both or either of the compounds in a larger concentration. Now from the IMP checkpoint, why am I calling this as a checkpoint? IMP acts as a checkpoint to direct the pathway of the GMP and the AMP. Suppose in the cell, the concentration of the GMP pathway, GMP, is very much higher. So it will direct the inhibition, or it will inhibit the pathway of the GMP synthesis, that is xanthylate ribose 5 monophosphate to the guanylate ribose 5 monophosphate. But it will not hamper the pathway of the AMP. Now suppose the contrarily the converse is true, that is AMP is higher within the cell. So what will AMP do? What AMP does is AMP inhibits the process or the direction of the synthesis of itself, but do not hampers the pathway of the GMP. This is how the allosteric feedback regulation happens. Beautiful, isn't it? So AMP and GMP beautifully incorporates each other, directly counterbalances or directs the counterbalance or reciprocal operation of each other's pathway. Right. So there is a reciprocity or counterbalance in the synthesis of the AMP and the GMP, guanylate ribose 5 monophosphate, adenylate monophosphate. Now you see right here that guanosine triphosphate is getting converted to GDP plus PI and liberating the energy that is necessary for the formation of the adenylate ribose 5 monophosphate. On the other hand, the ATP is getting converted to AMP plus PPI and liberating the formation energy that is helping the formation of the guanylate ribose 5 monophosphate. So why is adenosine nucleoside helping the formation of the guanylate or the guanine residue, and why is the guanosine nucleotide helping the formation of the adenylate residue? This is what the counterbalance of the reciprocity of the formation in a balanced manner for both of these guanylate and the adenylate nucleotides happens. Because if guanosine, suppose the guanosine means the pentose sugar plus the guanine base, the guanine nitrogenous base, helps in the formation of the guanylate ribose 4 phosphate residue. Suppose in the cell, in case of emergency, wherever the guanylate or adenylate is being needed, suppose in case of protein deficiency, what happens? If it would be the other case, that guanylate, the GDP is directing the formation of guanylate ribose 5 monophosphate. So GMP, which is getting accumulated within the cell in much larger quantity than the normal quantity, will inhibit the incorporation of the energy from the GTP, because the same residue or the same nitrogenous base cannot direct the pathway of itself. When the guanosine triphosphate, when the guanosine monophosphate is becoming higher, then the triphosphate molecules will automatically become higher and it cannot direct the pathway of itself. So it cannot assist the formation of the guanylate ribose 5 monophosphate. And the same thing is contrarily true for the adenosine 5 monophosphate. When the AMP concentration rises, the ATP concentration rises reversibly, and the ADP cannot help direct the pathway of the AMP formation itself. So in this case, the counterbalance is very much necessary. That is the either or balance, the counteractivity of both the energy providing molecules, that is the adenosine, which is the nucleoside of the adenosine adenylate ribose 5 monophosphate. The nucleoside of this AMP nucleotide is helping in directing the pathway of the GMP, right? And on the other hand, the guanosine, which is the nucleoside of the GMP, is helping in directing the pathway of the AMP. Because the same molecule, I'm telling you again, the same molecule, the same nitrogenous base, or the same compound which is having the same ring structure or the likeliness or the similarity, the similar compound cannot direct the derivatives of itself. Because when the accumulation of GNP or ANP rises, then reversibly, along with the phosphate group, it will help direct the formation of the ADP or GTP in some other pathways of the cell. And it's very inevitable or evident for the production of the passive or active processes, for the production of energy, rich compound and the cell signaling molecule. The PPI or the PI molecule will again combine with the monophosphate molecule and form the triphosphate. Either it's guanosine triphosphate or adenosine triphosphate. So it will form reversibly. It will form in a reverse pathway. It will form the guanosine triphosphate, the GDP molecule, for along with the GDP plus PPI or the AMP plus the PPI, whatever it is, right? So in this case, in that case, the same molecule, the same nucleoside cannot direct the pathway of itself. So the counterbalance is very much necessary for directing the regulation of the pathway to formation of the GMP, where the adenosine nucleoside is helping in direction, or adenosine nucleoside is participating. And in case of the adenosine, adenylate 5-ribose 5 monophosphate, or the adenine nucleotide, ribonucleotide, adenylate ribonucleotide, the GNP or the guanosine residue or the nucleoside is helping in directing this pathway. So you see how the counterbalance is beautifully measured in order to have a feedback regulation, that whenever either of these molecules are higher, the same molecule will not be produced, rather the other molecule will be produced, but with a derivative, but with a nucleoside of the molecule, of that molecule which has been inhibited to form itself. So this is all about how the allosteric feedback regulation of the de novo pathway happens in the cytosol, endoplasmic reticulum, and also in case of the Golgi complex. So this is all about the de novo synthesis of the purine nucleotides, the guanine and adenine. So if you like this video, please subscribe my channel positively to show your love and affection more, so that I can make more such videos for you people. And also comment down in the comment section below, and also let me know if you have any personal queries or doubts, and don't forget to hit the thumbs up button. Thank you.